Good afternoon, everyone. I'm Natalie Pipridis, and I'm delighted to extend a warm welcome to all of you joining us today for the SRA Webinar series. And once again, I'm very happy to see that we have a really good turnout, and I'm confident that our session will prove to be very interesting today and maybe even entertaining. I'm convinced that our must-attend event will provide fundamental perspective on the engaging landscape of biotechnology and breeding. So today I just want to start by acknowledgement of the country. So I would like to begin by acknowledging the Aboriginal and Torres Strait Islander people of this nation. We acknowledge the traditional custodian of the land on which SRA is located and where we conduct our business. We pay our respect to respects to ancestors and elders, past and present. So today's presentation will run for about 40 minutes and followed by a Q&A session. I would like to introduce our speaker, Professor Ian Godwin, whose presentation today is titled Adventure in Crop Biotechnology with Sorghum, Sugarcane's Less Complicated Sister Crop. Indeed, since I've been working on sugarcane, sorghum has always been the genetic model for sugarcane. Sorghum is the diploid closest crop for sugarcane, and a lot of the genome have been conserved between the two species. And until recently, we're still using actually the same chromosome numbering system for sorghum and to design the sugarcane chromosomes. Once again, I felt very privileged because despite his very busy schedule, Ian accepted without hesitation to give this webinar. Ian has 30 years of experience in biotechnology research, and since 1990, he has held an academic position in plant molecular genetics at the University of Queensland, serving as the director of the Centre for Crop Science in Quafi since 2019. His research involves the use of biotechnological tools for crop improvement with a major focus on the improvement of crop for food, feed, and bioindustrial end users, including biofuels and biomaterials. The Centre for Crop Science currently has 81 research staff and 54 PhD students, with an externally funded research budget of around 25 million in 2024. Ian is the Australian lead of the International Research Training Group for Accelerating Crop Genetic Gain with 3 German institutions, aiming to train over 50 PhD students in modern plant improvement technology. In 2019, Ian's popular science book, Good Enough to Eat, was published by the Royal Society for Chemistry. And in 2021, Ian was awarded the Medal of Agriculture by the Australian Institute for Agricultural Science. I still remember the first time I saw one of Ian's presentations at Trobag, and I definitely remember the last one, where Ian was an invited plenary speaker at the prestigious Plant and Animal Genome Conference in San Diego. And in front of more than 3,000 scientists, Ian was crouching up and down the stage to demonstrate how to walk under a very low polytunnel, assessing his sorghum trial back in Queensland. So I'm confident that today Ian's presentation will captivate you and will be memorable. So without further delay, Ian, the virtual floor is yours.
Thank you very much, Natalie. That's thanks for the kind introduction. And when you were saying all that stuff, yeah, I just thought to myself, well, I guess what that shows is I'm old because I've been around long enough to do some things. So where is my? Here we go. It's pretty exciting, isn't it? Oh, there we go. Are you seeing the correct view?
Yes.
Yeah. Okay. All right. Great. Okay. I did change the title a little bit because when I first put the title forward, I said it was sugarcane's less complicated and better looking little sister, but I think that was perhaps being a little bit too flippant. And I'm going to basically go on to the why am I working on sorghum, for example, what some of the things that sorghum can give us as geneticists. A little bit about what we use the biomass for, because most of my research has been on sorghum as a grain, because sorghum does have grain. Now, I've put there, perhaps flippantly, that it's quite novel. I mean, compared to sugarcane, it is novel, but realistically, most grasses around the world don't actually have grain that we eat. So having grain that humans eat is actually a quite novel thing for grasses. And then I'll talk a little bit about how some of our gene editing work and how can that help to improve sorghum for its end uses. So one of the reasons why I like sorghum is because it has a small genome, which is, there's a Circos diagram there from a paper we produced in 2013 with Emma Mace as the senior author, and it shows that although there are a few minor duplications around the place, those the 10 chromosomes of sorghum are very much like a true diploid, unlike the situation if you were to do the same Circos diagram with maize. There would be duplications happening all over the place. And I don't even know if you can do a Circos diagram with sugarcane, because I think it would actually be unintelligible just because there's so many chromosomes and things happening. Another thing that's really great for us is you can get through 4 generations a year, which means if you want to do any sort of genetics, you have the ability to really get through to F6 if you need to in not a lot of time. But as well as being a grain, it's a forage, it's a fodder, and it's a bioeconomy feedstock, which is growing in importance in many parts of the world. I want to talk a little bit about this because just to basically say that as Australians, we're very proud of some of the things that Australia is good at. Our cricket team, some of our sporting teams, the way we do really well in the athletics, etc., and swimming, and the Olympics. But actually, we've got an international reputation for doing drought really well. We are very, very good at drought, and it's not too long ago that we were in one of the worst droughts of our recent history. Cropping was really affected by that. Across most of Australia, we had bushfires everywhere. And then, of course, as we know, when it ends, it ended with floods. So the poems about Australia from 100 years ago are all very true. This is a picture actually of a sorghum nursery in Germany, because we do have some collaborations going on with Germany, as Natalie mentioned. And if you, going back to the whole drought thing, if you went to a conference in Europe 20 years ago as an Australian and started talking about drought and heat, they would sort of look at you blankly and say, yeah, that's, you guys are obsessed with this. It's not interesting. And now they're growing large amounts of sorghum, for example, because they're getting lots of drought and lots of heat. And they can, with sorghum, because it's a C4 photosynthesis plant, you can get more biomass with less water. It does have higher nitrogen use efficiency than maize, which is a major competitor. It definitely has higher heat tolerance at flowering, which means you can still get grain under conditions that crops like maize will fall apart. And you can get some pretty good biomass yields as well. Perhaps not as good as you can get with sugarcane. And it definitely has the disadvantage in that it doesn't return for as long as sugarcane. But if you irrigate it, you can get some pretty high levels of dry matter from sorghum. There's a picture here of one of the typical silage type sorghums. So, at first glance, you could look at that and think that maybe that was a sugarcane crop. But the biomass for sorghum is used for, when I say forage and fodder, they're different in the sense that forage is where the animal is actually eating it, grazing directly on it in the field. Fodder, it's usually harvested, often turned into hay or silage, and then eaten at a later date. Sorghum, in many places in the world, it's grown as a dual purpose crop. So the stover is just as important, or if not sometimes more important than the head. And beef, dairy, sheep and lamb industries use it in Australia. Some sorghums are actually used as specific break crops for soil pests and diseases because they have a number of root exudates, which, for example, will reduce nematodes, etc. And of course, it's used as a biofuel. We do forget that the grain is a major staple for about 700 million people. You can think about what you use as a staple, and most commonly it's flatbreads or porridges for sorghum, but beer is made out of sorghum in many parts of Africa. And of course, the famous baijiu drink that some of you have probably been lucky enough to try. And I don't want to be unfair about it, but from my point of view, baijiu is something that not only will you never forget the first time you taste it, you'll also never forget that you can still smell it the next morning when you wake up. But it is the ultimate in upvaluing something from a value adding point of view to basically distill sorghum and sell it for $250 a bottle to a large number of people. And in fact, for some reasons that are probably obvious to people who know me, this is one of my favorite books. Amy Stewart says, if we had a way to accurately tally the world's vast and complex drinking practices, sorghum is clearly one of the world's most imbibed plants. And most of that is actually baijiu. And although many people have never heard of it, have never actually even tasted it, if you go to China, for example, you can't avoid it. And I want to tell you that, in fact, as a single drink on an alcohol basis, the amount of baijiu that is drunk is the biggest source of alcohol imbibed by people, followed by vodka. And vodka is only about 60% of the amount of alcohol that's drunk by baijiu. Just a little plug about some of the things that are going on in coffee in particular at UQ, which are of relevance to sugarcane and sorghum. So we've got an ARC research hub for engineering plants to replace fossil carbon, which is about to be contracted in October this year, led by Robert Henry. An ARC training centre in predictive breeding, which is led by Lee Hickey. Also, we've got a CRC zero net emissions in agriculture, which has partnerships all over the world, but includes people at SRA. And we do have some current SRA projects and some more potentially pending SRA projects in coffee. And a number of those are actually capitalising on what Natalie said at the beginning, that sorghum and being a model potentially for sugarcane. But sorghum biomass is more than biofuel. And this is Jordan Pennells, who completed his PhD a number of years ago with myself and Darren Martin, who's in chemical engineer, and Alan Cruikshank, who was a sorghum breeder at DAF at Warwick and very sadly passed away last year. Jordan's whole PhD was looking at how you can process sorghum and whether sorghum biomass was likely to be competitive against other sources of biomass. And this is an engineer's view of the sorts of things you can do with biomass, but basically it was being broken down into nanofibers and then being reconstructed into nanopaper, which is a good way to test the tensile strength of the sorts of materials you might be able to make with sorghum. And one of his big comparisons, he did compare sorghum biomass with sugarcane biomass, with banana biomass, with various softwoods, spinifexes, and hardwood. And this particular comparison on this graph is just the tensile strength of sorghum versus sugarcane. But in fact, overall, sorghum nanopaper had a higher tensile strength, meaning it will give you more industrially advantageous outcomes than any of the other types of biomass. What, of course, wasn't looked at was really to much of a degree was the fact that Jordan did this with a sorghum, a sugarcane, a banana type, which was Cavendish, of course. And we know, you talk to a chemical engineer and you start talking about genetic diversity, and it just sort of washes over them with, why should we care about that? Well, the reason why they should care about that was there was huge genetic variation for different nanoparticle qualities from different sorghum lines, and we were able to demonstrate that quite a bit. Some of them were more suitable for water retentive hydrogels, and Darren himself has got projects going using hydrogels for all sorts of things, from cosmetic fillers for people to using these hydrogels as soil additives, particularly in protected cropping. There's also a project he's involved in with utilising these for electrode components in next-gen storage. Another interesting thing that we collaborated with people at the University of Hong Kong and at Cornell University, and we were applying gene editing to actually change some of the basic biochemicals that are made inside inside sorghum, but we were also doing that as a comparison with sugarcane as well. I just want to focus on the left-hand side, the still bean biosynthetic pathway. But one of the steps along that pathway is a thing called resveratrol, and resveratrol has been identified as, for example, being in things like blueberries and red wine as a strong antioxidant, which supposedly has good health outcomes. And so this team were really interested in saying, can we use sorghum or sugarcane biomass to actually produce large amounts of resveratrol? The problem with the resveratrol is it's largely it's largely turned into things like pinostilbene and pterostilbene by a particular enzyme. And so our aim was, can we knock out that enzyme and make sorghum that accumulates large levels of resveratrol? And the answer to that was yes, we could. We could really increase the still bean content of sorghum, particularly after wounding, and then by knocking out the sorghum SOMT, it led to actually resveratrol accumulation, really high levels of resveratrol accumulation. But you have to wound the plant. But from another point of view, one of the things that we're really focused on there is it's also been demonstrated that resveratrols are pretty good at reducing methane emissions from cattle. And how do we know that? It's because of a group of French scientists who thought, I wonder what happens if we feed the waste from wine grape production and we put that into cattle. And they showed that if it was a white grape variety, it had no effect on methane production whatsoever. But if they had a red grape variety, there was some of them, like the Shirazes or Syrahs, had a really positive effect on the reduction of methane. And they actually did a chemical analysis of that. This was mostly work done at INRA in France, and they were able to demonstrate that the resveratrol has a great effect on the methanogenic bacteria. It actually inhibits many of the methanogenic bacterium that live in the cattle rumen, which is quite interesting. So we're thinking, can we use sorghum? And because if you're going to turn it into hay or silage, you do have to wound it. Can we use this to really make large amounts of resveratrol in that? Unfortunately, in sugarcane, it doesn't. Doing these manipulations doesn't actually lead to any resveratrol accumulation at all. I'm going to spend most of the rest of my talk talking about grain, and I'll be particularly focusing on the grain. The major use of grain in Australia is either for poultry feed, predominantly for meat chickens, or it's for pork feed. But we've done a lot of work with the poultry industry, and I'm going to outline some of that. So most of the sorghum hybrids in Australia produce about 10% protein, and meat chickens in their diet require at least 22 to 24% protein in their diet. So what is done in the Australian industry and most industries around the world is they use soybean meal to bring that protein content up. What the industry would really like would be, are we able to produce a cereal that they can buy more cheaply that has an increased protein content and an increased protein digestibility, that means they can use less soybean meal in their diets and therefore make cheaper diets for chickens. They also want to ensure that they get a low feed conversion ratio. Now, the feed conversion ratio is the thing that the poultry industry uses. It says to get 1 kilogram of chicken, how many kilograms of feed does that chicken need to eat? And in most commercial industries, it's around about 1.5. So 1.5 kilograms of feed is required on a per chicken basis to produce 1 kilogram of chicken, live weight, not necessarily meat only, but live weight of the chicken. To be able to do some of these things, we needed to have not just a sorghum genome, but a sorghum pan genome. Anyone who's used the basic, the reference genome of sorghum, that reference genome is a thing called TX623. And it was a widely used sorghum hybrid pairing in the United States and other parts of the world as well. So that was why that was actually selected to be the reference genome. We now know if you wanted to make the worst choice to make a reference genome, you would start with BTX623. And the reason for that is it's got less genes than every other sorghum, which is quite interesting because this work was led by Yongfu Tao and Dave Jordan and Emma Mace, able to show that in cultivated sorghums, gene number varies from 32 to 36,000. So some cultivated sorghums have got 4,000 more genes than others. And probably that presence-absence variation is a big driver for the heterosis we see when we make hybrid sorghums. For our tissue culture and transformation and gene editing, we all use another line called TX430. It came from exactly the same breeding program as TX623, but it's got 900 more genes than TX623. Now, one thing that I want to point out about, of those 900 more genes, very few of them are actually duplications of the same thing that TX623 has. They're just genes that 623 doesn't have, which is quite an interesting situation to be in. We also need a transformation system, and this is Guochuan Liu, who, as part of his PhD more than 10 years ago now, developed a really good transformation system and then started working on various projects to increase protein digestibility, content, and grain size. In the past, we always used microprojectiles. We have switched to agrobacterium. If you want to know why, I can give you a long, convoluted answer, or I can give you the short answer. The short answer is our Institutional Biosafety Committee said microprojectiles lead to gene fragmentation. We're not going to believe you if you don't say there's a transgene there because we believe there'll be fragments. You have to use agrobacterium. So now we use agrobacterium for all of our transformations. To do gene editing, we started our gene editing experiments predominantly using things that people had used in rice. They didn't work terribly well with sorghum. Karen Massel, as a postdoc, did some work on actually using some sorghum U6 promoters, and they had huge increases in improved editing efficiencies. And we now know that those U6 promoters give really high levels of editing efficiencies in sorghum, barley, and through some collaborators with maize as well. We have now edited almost 50 different genes in sorghum, all in TX430. A couple of them are genes that don't exist in 623, which is interesting in itself. I'm going to mostly focus on the things that are involved in feed grain protein and digestibility as well. One of the genes we knocked out is a protein foldase gene, which we use that particular protein foldase gene so that the proteins that are stored in the sorghum grain would be folded in a different way. Our prediction was that they would have fewer disulfide bonds and therefore be more digestible. And this is where the serendipity of science comes through. They always also have a higher kernel weight, which was totally unexpected, and they have a higher protein content. So some of these lines have 15 to 16% protein, and you'll recall that the standard sorghums we grow have about 10% protein. So this is a massive increase in the protein content of the grain. That was done under an ARC project. We then got a GRDC project called Better Sorghums, and the whole aim of that Better Sorghums project was actually to do feed trials, and with our with our colleagues at University of Sydney, to do some chicken feeding trials. So we were going to feed these these improved grains to chickens to see, do they actually work in a chicken. It was important to get these things out into the field, and the reason for that is because you know, that's where it all happens. But we also know that growing, you probably have the same issue with sugarcane. Excuse me, got a bit of a dry throat there. My apologies. We can't get high enough photosynthetic radiation in a PC2 glasshouse for sorghum, so it always grows better in the field. Also, the roots grow so quickly that even in a really large pot, the roots have reached the bottom of the pot in about 10 days, and the plant starts to say, you know, oh, we've hit a soil barrier here, so set up the plant to be somewhat smaller. So you don't get the same yield per plant, and you never will, because you're never actually getting proper photosynthesis as far as a C4 plant is. So one of the things we were able to see, our GS3 knockout lines, which is a grain size gene, had even larger grains in the field. Fantastic outcome, and had higher protein contents, which we'd never actually measured in the glasshouse. We did have another construct, which is called DEP1, dense and erect panicle, which gives you higher grain numbers in the field. But for some reason or other, that change we made to plants made those plants very susceptible to heat at flowering and grain fill. And in the second year of trialing, we had 17 days where it was 35 degrees or above, and these lines, they just didn't yield. As you can see, we did this under a net because the OGTR said we had to, because birds would steal our grains and spread them all over the world, and we had to bag every head because, you know, sorghum pollen was going to wreak havoc. So this particular trial that you can see on the right, we bagged 60,000 sorghum heads, and that was in that second year where we had 17 days during grain fill, well, during flowering and grain fill, that were over 35 degrees every day. So that was a lot of fun. We dealt with it by starting bagging at about 4 o'clock in the morning. We were able to make some really gross changes in endosperm structure. So the Foldase knockout I talked about earlier gives us mostly flowery endosperm. The GS3, the grain size one, and I'm focusing on these because these are the ones that ended up performing best in the poultry diets. They had a totally different change, and they had hardly any flowery endosperm. They had mostly corneus. But both of those lines were delivering us 15 to 16% protein on a dry matter basis. And that was from field trials across 3 years and in different places, because we also did field trials at Cecil Plains. The poultry industry at the beginning actually told us that they wanted a white sorghum like Liberty. It's the only white sorghum in Australia. They believe that their chickens do better with that particular line. They wanted a cheaper source of protein, which was going to be sorghum potentially. They wanted to have live weight gain, the higher the better. What they would love would be the same feed intake with higher digestibility and get more live weight gain. And I've already talked about the feed conversion ratio. They really want to get the feed conversion ratio as low as they can. And they've always said that they get a feed conversion ratio, the best feed conversion ratio is from Liberty, which hardly anyone grows because it often gets downgraded for quality reasons. So these were this was the team from the Poultry Research Foundation at the University of Sydney, Sonia Liu and Peter Selle. The question that we were asking was, can sorghum protein perform in commercial poultry diets? And our comparison was really using commercial hybrids where you had 10% protein and you had to use more soybean meal, or our better sorghums, which had 15 to 16% protein and you needed less soybean meal. And really the question was, if we make diets that have the same amount of protein, but more of that protein comes from sorghum, do you get equivalent growth, or does this have some sort of deleterious effect? The outcome actually was that, and you can see on the right-hand side, these basically the feeding trials went for 35 days. After 35 days, the chickens are ready to go to the Oktoberfest. So what we were able to demonstrate was with those higher protein sorghums, they had 16% protein in the diet. So you would use about 100 kilograms less soybean meal in the diet. Now those figures vary from time to time. At the moment, the soybean meal is around about $1,200 per ton imported, and 100 kilograms more sorghum in the diet. But also I'd have to say that the sorghum's now more $350 a ton. So you can see that the monetary comparison changes depending on the relative prices of those things. But what we were able to work out that using these feeds, you could get about a 24 to 28 cents saving per chicken just in those feed costs. And Australians eat about 2 million chickens per day. So on a per annum basis, this is many, many millions of dollars saved to the poultry feed industry. If we can deliver these same benefits in commercially adapted hybrids, that's the point I really want to stress here. We have done this in one sorghum line. The next step that we're going towards next year is to develop these same things in some of the Australian adapted hybrids to see whether we can also get 15 to 16% protein. But in reality, even if we only increase protein to 12 or 13%, the industry would be really interested in that. I'll just talk a little about some of the other things that we're doing with sorghum. So the cafferins are the seed storage proteins of sorghum. And we know that particularly in West Africa and particularly around Nigeria, a lot of the lines that are grown have a naturally occurring beta-cafferin knockout. So they actually don't have a functional gene for beta-cafferin. Now, beta-cafferin is one of those cafferins that is highly disulfide bound, which makes it really resistant to protease digestion for monogastrics, so us, pigs, chickens, etc. Interestingly, we found that all of the lines that had this mutation, they had exactly the same mutation. It had come from the same evolutionary background, and it had been selected for in land races by people who make sorghum porridge, and a particular sorghum porridge that in northern Nigeria people prefer to eat. They say it tastes like silk on the mouth. It is a porridge called Ogi Bawa, and we were able to show that all of the lines used for Ogi Bawa have no beta-cafferin. We also know that the lines with no beta-cafferin tend to have higher protein, a little bit higher protein, and the protein is definitely more digestible. So we thought, well, this is a low-hanging fruit. Let's just knock out the beta-cafferin gene in TX430, and we're going to get higher digestibility and possibly even higher protein. Oops. And the outcome was it didn't really change much at all. There was hardly any change in grain size, hardly any change in grain weight. There was no change whatsoever in protein content. And this is Karen Massel, who led a lot of this work, who now leads her own team at UQ. There were no quality changes at all. And in fact, the beta-cafferin knockout lines may have actually been less digestible, particularly in chymotrypsin. So we couldn't work that out. It didn't make any sense at all. But what we do know now is that after doing proteomics, so you can see here that the beta-cafferin is totally knocked out. There's no gene expression, little or no gene expression whatsoever. But what does happen is when we knock out beta-cafferin, it actually triggers an increase in gamma-cafferin. And gamma-cafferin is the other highly disulfide bound protein because it's got a higher level of cysteine residues. So the gamma-cafferin increased in these knockout lines and actually had a deleterious effect on digestibility. So instead of getting more digestible sorghums, we were actually making them worse. So we then thought, okay, well, what are we going to do now? And the obvious thing was we could go and spend a lot of time working out why that beta-caffeine thing didn't work, but it sort of pointed to gamma-caffeine as being that one is a key one. Let's knock out gamma-caffeine. Now, there is some literature, older literature, that says if you knock out gamma-caffeine, sorghum won't be able to make protein bodies and it will actually be seed lethal. It turns out that that's not actually the case. You've actually got the knockouts actually give us an increase in digestibility, and it also works in cattle. So we've been able to look at some of those in cattle as well. And I seem to have somehow or other duplicated a few slides here, so I'm just going to shoot through. Apologies for this. What we know about some of the lines that have given us the best outcomes in the poultry, these foal days knockout lines, is that they have a high level of protein. And it seems what the big change is, and you can see these red arrows. On the left, you've got TX430, which has got a very small level of what's called the peripheral endosperm, which is almost entirely protein bodies. By knocking out those, by knocking out the foldase gene, for some reason or other, the peripheral endosperm is much larger, and that's what drives the higher level of actual protein in these grains. In the field, we are able to show that quite a few of these lines not only have more grain, but they also have larger grain. And from our field trials in 2019, we grew the selected favourites to actually to go into our feeding trials, as I discussed. Now, what this graph is showing us here, you'll see on the right-hand side is TX430, and TX430 just gets 0 for everything, because what we're doing here is comparing what was the change in either protein content in the purple bar, the 1,000 kernel weight in the red bar, or the seed number in the blue bar. And you'll see we selected these lines to go into the poultry feeding. We did a couple of those ones we got rid of because they were the ones that failed in the heat. And these were the lines that we ended up taking forward for our second year and growing them again in a larger experiment to actually get a bigger amount of grain that we could use for poultry feeding. We could do the same sort of thing with pigs, but the reason why we've really worked with the poultry industry is because if we can grow 70 to 75 kilograms of a particular line, that's enough for them to do a really good replicated feeding trial. If we wanted to do that with the pork industry, you'd actually be talking about 700 kilograms. And to grow 700 kilograms of grain that you're growing under containment conditions under a DIR license is a big deal. I just going back to the broiler feeding trials, just to the trials that they were done would take 320 day-old chicks and feed them for day 35. And our colleagues in the University of Sydney called these called day 35 kill day, which to some of the plant scientists in our group made them a little bit squeamish. So they kindly changed that to data collection day for us to make it a bit more palatable. I'll skip through that data because I've already talked about that. And I just wanted to finally end up on the whole story of what happened with our gamma-caffeine knockouts. The gamma-caffeine knockouts are really, really promising for us. The knockouts, they're totally normal plants. They yield in a fairly normal way in most cases. But when we do the digestibility, particularly in the chymotrypsin, which is the digestibility that happens post-stomach, you're actually getting double the amount of protein is digested. So what we're really aiming to go for next is to try and get some combinations of, for example, if we can combine the foldase knockout with the gamma-caffeine knockout, we believe we'll get higher protein, and that protein is going to be really, really digestible and therefore is more likely to be useful for poultry and pig feeding in Australia. Internationally, it will probably also change the digestion profiles of sorghum for humans, because in humans where sorghum is eaten as a porridge or as a flatbread, the cooking process interacts badly with those cysteine residues in the disulfide bonds and tends to make the protein largely indigestible, which means that people who are eating a lot of sorghum in their diet aren't actually getting the metabolizable energy that they should be able to get. That was the testing, but I just wanted to show you here that in monogastrics, we've been able to increase the digestibility. We've been able to do the same in ruminants, and a really good outcome that we hadn't really foreseen was you're getting lower methane production in those cattle as well. So we've got a lot of promising lines of research that we're going for. Our particular focus at this moment is to work with seed companies to be able to develop these same types of changes to the grain in their hybrids. To do that, we will need to be undertaking gene editing in their elite backgrounds, which will be a new challenge for us, but it's going to be an exciting challenge. But I expect once we've got all of the agreements settled out with that, we would hope to actually be having hybrids ready to do some field testing within about 3 or 4 years' time. So I'll just finish with this, that genomics and gene editing plus some physiology gives us better sorghums. And thank you for spending your Friday afternoon with me.
Thank you, Ian. Really a great presentation. I'm a bit jealous because, you know, sugarcane is so much more complicated, like genetically. But anyway, it's time for question now. So if you do have a question for Ian, you can type it in the chat or raise your hand using the hand button, and then it's at the top of the bar, and then we'll unmute you. So then you will need to unmute your microphone to ask a question when prompted, and then mute it again. Does anyone have a question for Ian? Oh, well, so at the moment there's no question, but I've got a few. So as usual. So Ian, I was just like that, what you said about the French people, like, you know, and the wine things, is it, I thought like, oh God, those French people are so smart. Do you,
Obviously.
does that work for everyone? Like, I mean, that was tested on like cows or cattle, obviously.
Yeah, it was tested on dairy cattle. They just wanted to see if it had value as an animal feed. The outcome was, once they did the sums, unfortunately, was that on an annual basis they would never have enough of the waste products to really turn into anything that was commercially viable, and that was the problem. But it did enable them to look at the components of it was, you know, an interesting comparison between, you know, if you've got a Chardonnay waste versus a Syrah waste, that you were getting different outcomes, and then that helped them to work out that, well, there's something in the skin of the grape, that in those red grapes, which is actually giving you the anti-methogenic bacteria activity. And they tested a number of things, but it turned out that resveratrol seemed to be the one that answered most of those things. Now, all of that resveratrol work was actually done in vitro. So again, you know, scaling that out to production level animals may be a challenge. Another thing that, you know, having worked with some people working on cattle rumens, what you don't know is you might develop something that it changes the rumen microflora population, and you get a decrease in the amount of methane produced. However, does that continue over time, or does that, you know, are you selecting for specific methanogenic families that can then build back up in the rumen, so that maybe you're only getting an effect for a couple of months or something or other until the rumen population changes and methane production starts again. Those are the sorts of things that would have to be answered much more long term than first, can we develop this in something like forage sorghum?
Okay, thanks. Oh, also, I just wanted to tell you, like, we do have circus in sugarcane, but I think, like, I mean, Karen can correct me. When I do some, I just have one chromosome at the time, because we do have like 12 or more copies per, so yeah. And maybe Karen knows if there's like one, like global ones, but yeah. And we always like, have like the comparison with sorghum. And I feel like really bad now that I use 623 all the time, and now like you're telling me that it doesn't even have all the genes. So but anyway, we got a question here, so hang on, just if I could get my mouse onto it. So Alexander, this is so
yes, thank you very much, Ian, for the presentation. Just back to the trial with the chickens and the sorghum. What was the end outcome of that?
So the outcome was that the feed conversion ratio was as good or better. The live weight gain was not statistically different. There didn't appear to be any abnormalities in one of the diets. The chickens on one of the better sorghums appeared to have a slightly higher level of fat on the breast meat. Otherwise, there were no detectable differences. And so that's why we're sort of going further. But as I've said, the genetic background itself could be problematic, in the sense that if we try this in other hybrid parents, are we going to get those same changes? And I would suspect that we'll probably get similar changes in some of them, but whether it works in all, I can't really predict. Yeah.
Right.
I'd be surprised if it worked as well in all of them. It just seems a bit too good to be true.
Right. Yeah. Thank you very much.
Thank you, Ian. Any other question? So my other question, it's back to the TX430. So you said they've got like 4,000 more genes than 623.
Well, it's got about 900 to 1,000 more genes than 623. So the 430 genome is now, it's available, publicly available, as is a couple of others, and another one of them is actually a sweet sorghum called Rio, and that sorghum has quite a few, even more genes that 623 doesn't have.
But so what's the difference in like biomass or like grain between like 623? Like, I mean, does it make a big difference? Like where does these genes have any consequence?
Yeah, so it's a great question. So the people who've studied sorghums, because sorghum came into cultivation, it was domesticated in sub-Saharan Africa, probably around somewhere like Sudan, Ethiopia, that type of region. But there's different separate domestications also in Southern Africa and in Western Africa. And those sorghums are quite different. So they actually have given them what they call a racial structure. And TX430 is very much like a cordatum, which is a sort of east sorghum type, whereas 623 is more like a Dura sorghum. So they're actually selected, and those races were domesticated in different parts of Africa. So it's probably more just a reflection of those separate domestication events that really drove those differences in gene number. Because at the time, of course, no one was deliberately making hybrids. Everything was grown as a land race, an open pollinated land race.
Yeah, and that makes sense. I've got a question for George. So, George.
G'day, Ian. How's it going? Nice to see you, and thanks for the presentation. It was great.
Well, thanks, Stuart.
And I like the way you bring alcohol and food into your presentations, but a bit disappointing that you didn't talk about rum. Anyway,
I can give you a bit of information about rum if you want some.
Maybe. So, like you mentioned, you've been working in GM and now gene editing for a long time. So what sort of changes have you seen in the regulations or the regulatory bodies in terms of having to trial these in the field? And where do you think that's going to go?
It's a great question. So Australia overall is it hasn't said no to gene edited crops in the sense that it hasn't said, like, for example, New Zealand and Europe did, that the gene edited plants are actually GM. So we're a little bit more relaxed than that, but we're a long way behind, for example, jurisdictions in Canada, the US, Japan, etc., who are allowing quite a lot more gene editing going. Interestingly, I mean, a lot of people who have followed this know that Europe is currently debating whether to allow gene edited crops to be grown as non-GM. The pressure on a lot of governments has been seed companies and farmers are saying, we really missed the boat by saying we were going to refuse to have GM. This was not a good outcome. So please don't make the same mistake with gene editing. And so there is a loosening up. And as we know, one of the countries that's had the strongest anti-GM line has always been New Zealand. New Zealand's actually now talking about changing its legislation on gene-edited crops to actually make them more liberal than our rules in Australia. They're actually following what more along the lines of the Canadian model. And the Canadian model doesn't really look at the technology per se. It actually looks at the outcome and says, does this outcome that you're going to grow and eat, are there any additional risks, regardless of how it was made? And if they decide that tomato or that potato has no more risks or may actually have benefits, they're going to say no restrictions on it. On the rum topic, I did say to Natalie, because I know you sugarcane people are very proud of your rum, that I've had rum made from sorghum, from the actual sorghum stem and leaves, in both India and China. And firstly, I'd say I'm surprised I didn't end up going blind. And secondly, I'd say it tasted just like Bundaberg rum after you put 5 banana skins in it and let them sit for about a week. Yeah, so it's pretty bad.
Yeah. Okay. Yeah, I finished, George, or are you? Okay.
Yeah, so you mentioned just now that New Zealand that were very anti-GM or actually pro gene editing plants now. They got on the front foot with it. It reminded me of something happened several years ago with Japan. They were very anti-GM, and they actually contacted us and wanted to see whether they could grow some GM with it for them to test their plants because they couldn't grow them in Japan.
Yeah.
So it's really interesting to hear that they're going ahead with the gene editing plants. It's good.
Yeah, and similar things happening here because the industry and ag research and people like that in New Zealand are starting to get excited about the fact that, hey, we'll be able to actually do something with this. So they're all doing their field trials now in Australia because they can, and they don't want to be in a situation where they're sitting on something for 3 years waiting for the legislators to allow them to do their research. So they want to be in a better position.
Interesting. Thanks, Ian.
Ian, you've got a question from Al. As sorghum is easier to modify than sugarcane, should we grow a sweet sorghum to produce sugar and bioenergy commercially?
Yeah, another great question. So the sweetest sweet sorghums I would say slightly less competitive overall in terms of the amount of sugars you will get compared to the best sugar canes. That's as percentage of sugars. But the biggest problem with sorghum is that sorghum won't give you predominantly sucrose. It'll give you about, on average, maybe 50% of the juice is sucrose, and the remainder is usually a mix of fructose and glucose, which don't crystallize in the same way. Hence, sugar type energy going into things like biofuels, etc., or as animal feeds, no problem. But if you wanted to use it as an alternative to, it makes good syrups. It doesn't make crystallized sugar without a lot of intervention. So it's never going to compete on that front.
Thank you. You got a question from Ian Biggs. Do you think sorghum will have greater potential for these specific metabolites or as a whole grain?
Yeah. Look, that's another great question. One of the, as a whole grain, sorghum is generally regarded as a very healthy grain. For Western diets, one of the health outcomes is that it's more slowly digestible, which can be a very good thing. It may not, and actually potentially some components of it are indigestible, which might be great for some Western diets. It's not ideal if you're in sub-Saharan Africa. I think what is happening a lot in the US and in Southern Africa is people are, for example, growing sorghum lines and then actually extracting bioactive compounds from it. So they're using it more as a source of ingredients rather than people eating the sorghum. I mean, much along the lines of the canola that came out with the high omega-3. No one's buying bottles of high omega-3 canola oil, but you can go into a pharmacy in the US and you can buy capsules with the high omega-3 canola oil, and that's predominantly because you make more money out of that. The value adding means per hectare you're making a lot more money out of that. In saying that, 70% of the omega-3 canola is actually fed in aquaculture to fish, because it can be a replacement to the natural omega-3s that they would get in a natural diet or from eating other fish. And we have finally worked out that it's not a good idea to go and harvest a lot of anchovies and sardines and herrings and then grind them up and feed them to prawns. It's not very good for the circular economy. Did that answer your question well enough, Ian?
Well, I'll let you know when you write back.
Okay, no problem.
There's a comment from Lauren saying, great presentation. This is a learning curve for sugarcane research. Yes, and indeed, this is what we wanted to have you. I like I said, I'm quite jealous about the 20 chromosome compared to our 120 chromosome. But there was something you saying at the start about retunability, that
yes.
Sorghum doesn't have, I mean, I don't know how many times sorghum returns, but because sugarcane, like it's a big cost of whatever. So do you think that, I mean, first of all, how many times do you return sorghum, and what's the, if there's something to explore there in like the genetics to improve or to change the returnability in sugarcane from the sorghum?
Yeah. So there are projects around the world, including one potentially going on in Australia, about making sorghum more perennial, but they're largely focusing on, because there are sorghum relatives that have rhizomes and they effectively overwinter as rhizomes. So there are moves afoot, for example, to see, can we improve the perenniality of sorghum with those rhizomes. Now, where sorghum in Australia is ratooned, it's most commonly in the tropical areas because it usually gets killed by frost or cold weather otherwise. So central Queensland is where some ratooning occurs and grazing on that. There's been some research about just ratooning within the cropping season because you can get harvest within about 4 months. So if you plant early, like in August, you can do a harvest around Christmas time, and then you've still got time to get a second harvest of grain. But what on the coastal areas, it's really an absence of breeding for foliar diseases. That's the big issue. And you know, sugarcane people, I suppose, in the tropics, a lot of breeding effort has gone into overcoming potential foliar diseases, and that disease burden in sorghum, if you're using it in high rainfall areas, usually means that the sorghum effectively dies out from the disease burden rather than its ability to perennialize.
All right. There's a comment from Ian, back to you. So close. Work by Fricke found something similar for sorghum versus sugarcane in the Mosman region. You did find disease inhibited and early flowering inhibits yields of the sweet sorghums.
Yeah, that's true. So a lot of our sorghums in Australia are photoperiod insensitive, which means if you've got a line that is going to flower in 4 months, it's going to flower in 4 months regardless of what the day length is. You can, I mean, sorghum is naturally photoperiod sensitive, so you can go for photoperiod sensitive types that will, you know, well, much like sugarcane, will only flower once during the year, and it will be quite late in the season, and they're the ones that are going to give you the highest biomass.
All right. Well, thanks again, Ian, for presenting today. I think like it was really informative, and I will have more questions for you, but I'll just wait when I see. So thank you all for your attendance too. I hope you have enjoyed today's webinar and that you will join us for the next SRA webinar coming soon. So for information on the webinar, go to the events page of the SRA website. And then the webinar will be posted as well. Do you have any last word, Ian?
No, just thank you for having me, and thank you to the audience for giving up their Friday afternoon to listen to someone talk about something other than sugarcane. But it's been great, and thank you for inviting me, Natalie.
All right, thanks, Ian. Have a great Friday and have a great weekend. Bye.
Okay, bye.